Hello everyone, welcome to Aesthetic Food Tech. So in this video, I am going to discuss about classification of protein. Protein can be classified based on structure, composition and function. Classification of protein based on structure. Based on structure, protein is classified as fibrous protein and globular protein. Fibrous proteins are very strong and tough, and they are water insoluble, which form long fibers or sheets. Secondary structure is the functional structure of fibrous protein. Example, keratin. Globular protein. Globular protein have a spherical or globular shape. That is, protein structure is folded to produce a globular or spherical shape. Tertiary structure is the important functional structure of globular protein, and they are soft compared to fibrous protein. Example, myoglobin, hemoglobin and enzymes. Classification of protein based on composition. Based on composition, proteins are classified into three: simple protein, conjugated protein and derived protein. Simple protein. Simple protein is composed only of amino acid. They may be fibrous or globular. On hydrolysis of simple protein, we get amino acids. Simple protein have simple structure and organization. Based on solubility and heat coagulability, simple proteins are classified, and they are albumin, globulin, gliadin, glutenin, albuminoids, histone and protamine. First one is albumin. Albumin is soluble in water and also in dilute salt solution. It is coagulable by heat. Example, plant albumin, that is legumes and leucosin. Leucosin is present in cereals. Animal albumins include ovalbumin in egg and lactalbumin in milk. Second one is globulin. Globulin is insoluble in water and soluble in dilute neutral salt solution. It is coagulable by heat. Example, ovoglobulin in egg. Third one is gliadin. Gliadin is insoluble in water, salt solution and absolute alcohol. It is soluble in 50 to 80% ethanol. Example is wheat gliadin. Fourth one is glutenin. Glutenin is insoluble in water and salt solution and soluble in dilute acids and alkalis. They are coagulable by heat. Example, orizanin in rice and wheat gluten. Fifth one is protamine. Protamine is soluble in water, dilute acids and alkalis. It is a small molecule. Sixth one is histones. Histones are rich in histidine and arginine. These are amino acids. That is, histidine and arginine are amino acids. And also, histones are soluble in water, dilute acids and salt solution. But it is insoluble in ammonia. Example, histones of nucleoprotein. Seventh one is albuminoids. Albuminoids are fibrous proteins and they have low solubility and greater stability. They form supporting structure for animals. Example, keratin in hair and nail. Conjugated protein. Conjugated proteins are simple protein combined with a non-protein group called prosthetic group. The protein part is called as apoprotein and the entire molecule is called as haloprotein. Examples are first one, nucleoprotein. That is combination of simple proteins like protamine and histone with nucleic acid. Example, nucleoprotamine and nucleohistone. Second one is mucoprotein or mucoid. That is combination of simple proteins with micropolysaccharide. Example, beta-ovomucoid. Third one is glycoprotein. That is combination of simple protein with polysaccharide. Example, immunoglobulin G. And finally, fourth one is chromoprotein. That is combination of simple protein with a colored substance. That is, here a colored substance is the prosthetic group. Example, hemoglobin. Derived proteins. Derived proteins are proteins derived by the complete or partial hydrolysis of proteins by the action of acids, alkalis, or enzymes. There are primary derived proteins and secondary derived proteins. Primary derived proteins. Primary derived proteins are protein derivatives formed by causing light change in the structure of protein molecule. Peptide bond is not affected in this. Example, proteins, metaprotein and coagulated protein. Proteins are insoluble products formed by the action of water, dilute acids and enzymes in protein. Metaproteins are formed by the action of acid or alkalis upon protein. And finally, coagulated protein, which is formed by the action of heat or alcohol on protein. Secondary derived proteins are formed by the hydrolytic cleavage of peptide bond and protein molecule. They are grouped as proteoses, peptones and peptides. Proteoses are the hydrolytic products of protein. They are water soluble and heat coagulable. Peptones are the hydrolytic products of proteoses and they are water soluble and non-coagulable by heat. Example, products that is obtained by the digestion of protein in the body. Finally, peptides. Peptides contain small number of amino acids linked together by peptide bond. They are water soluble and non-coagulable by heat. Example, dipeptide. Classification of protein based on function. First one, structural protein. That is, protein that protect or strengthen biological structure. Example, collagen and elastin. Second one, enzymatic protein. That is, highly specialized protein that act as biological catalyst. Motile protein. That is, protein that function in contractile system. Example, actin and myosin. Transport or carrier protein. That is, proteins that transport iron or molecule in the body. Example, lipoprotein. Nutrient storage protein. That is, proteins that provide nutrients and store ions. Example, casein and ovalbumin. Defense proteins. That is, proteins that defend against other organisms. Example, antibodies. Regulatory protein. That is, protein that regulate cellular or metabolic activity in the body. Example, insulin and growth hormone. Toxic protein. That is, proteins that destroys enzymes. Example, bacterial enzymes. Egg protein. Egg protein contain two type of protein. First one, egg white protein and second one, egg yolk protein. Milk protein. Milk protein contain two types, that is, lactalbumin and lactoglobulin.